Hello and welcome to yet another informative segment in CliniLaunch Knowledge Pop. In today's video, let's learn about the most crucial part of clinical research, that is, what is blinding, what is bias, and why it is used in clinical trials. Let's start today's video with an interesting story of first ever clinical trial reported in the history of clinical research. It was back in 1747. The British sailors were suffering from a strange disease called scurvy. They were always fatigued, they had swollen and painful joints, their gums were red and bleeding and often lost their teeth. The severity of the disease was such that many people had lost their lives too. James Lind, who was a Navy surgeon, observed that the disease was seen in soldiers who were there on the ship for a long time and their diet consisted of salted meat and boiled biscuits. But James Lind did not have any previous reports to know what was really causing the disease and how to cure this. So, Mr. Lind started an experiment which goes like this. On 20th of May 1747, James Lind selected 12 soldiers and randomly divided them into 6 groups. Then he added an additional thing in their diet. One group received vinegar, one group received elixir of vitriol, which is the sulfuric acid, and another group received regular seawater, and one group received oranges and lemons in their diet. After 6 days of the experiment, the group which received oranges and lemons showed drastic improvement in their condition and they are fit for duty. This study is considered a breakthrough in the field of clinical research even today because of the systemic way it was conducted. This is a classic example of randomized clinical trial which laid the foundation of today's trial design. Paying tribute to James Lind on World Clinical Trial Day, which is on May 20th, let's dive into today's topic. So let's see what is blinding. As the name suggests, blinding means something you can't see or unknown. A blinded study design is a research method where one or more parties involved in the trial are kept unaware of certain details about the experiment. Now I want all of you to imagine you're having your birthday party. Birthday calls for gifts. At this stage, you don't know what kind of gifts you are going to receive. This is called blinding. In clinical studies, there are 3 main types of blinding: single-blinded study, double-blinded study, and triple-blinded study. Let's see what is a single-blinded study in detail. In a single-blinded study, only the participant or a group of participants are not aware of the medicine they are receiving. They may be in the experimental group receiving the investigational product, or in short IP, or they may be in the control group receiving the dummy drug or a placebo. If the parameter of the trial is dosage, then the participant wouldn't know if they are receiving the standard dose or the trial dose. Basically, in single-blinded technique, only the participant is unaware and the researcher would know the drug he is administering. Let's come to your birthday situation. Now, when you receive a gift, you don't know what is inside the package. But the person who is giving the gift would know what it is, right? This is called single blinding. For example, if a participant knows he is receiving a saline injection instead of a painkiller injection, he would react differently than if it's unknown to him. In other case, if participant knows he is receiving a new drug, then he may be very conscious about the effects. Both will lead to discrepancy in the trial results. Next is double-blinded study. It is the most common type of blinded studies. Here, neither the participant nor the researcher will not know to which group the participant belongs to. To keep it in simple terms, the participants are randomly divided into groups and the investigational product and the placebo will look similar in their package with their batch number written onto them. If it is an injection, they both will be in the same kind of syringes. If it is a pill, the both placebo and investigational product will look same in shape, size and color. If it's a drop, they both will be in the same kind of vials. Here, both the parties are unaware of who is receiving the investigational product and which group is receiving the placebo. Let's come to the birthday party situation. If your friend has sent another person to buy the gift for you, then you will also not know what is inside the package. Your friend will also not know, but the person who bought the gift will know what is inside the package. Similarly, a separate team a senior researcher, or a software system would know which group has received the real medicine and which group has received the placebo. For example, if I am testing a drug for depression, then the patient, if he doesn't know if he has received the real medicine or a sugar pill, then it's easy for the researcher to conclude if the improvement in the condition is psychological or physiological because of the medicine. If the researcher also doesn't know about the drug being administered, then he will not change his questionnaire, his extent and detail of the examination from person to person, resulting in unbiased results. Then comes triple blind study. This is the most rigorous form of blinding, where even the researchers analyzing the data are unaware of the treatment groups. This is less common because it requires an extra layer of complexity in managing the data. But it is involved in certain situations. It's like on your birthday, you don't know what is the gift, your friend don't know what it is, and the person who bought it also doesn't know what it is. Maybe he asked the shopkeeper to pack it. For example, if there is a study on a new surgical technique, might involve conducting a sham surgery procedure which mimics the real surgery but doesn't have any actual intervention. A separate team is responsible for assigning the patients into real surgery and the sham surgery, but the team analyzing the post-operative data wouldn't know if the patient belonged to which group until only after the analysis is complete. So why use blinded studies? Blinded studies are essential for obtaining the reliable and unbiased results in clinical trials, and they help in controlling placebo effect and observer bias. What is placebo effect? Participants who believe they are receiving an active treatment might report feeling better even if the treatment itself has no effect, meaning they might have received a placebo. Blinding helps in reducing the influence of these expectations. What is observer bias? The researcher might treat the participant differently or may interpret the responses in a biased way depending on whether the participant was in the experimental group or in the control group. The doctor may not examine a system, may not order for some investigation, or may change the questionnaire if they knows the person has received the real drug or the placebo. Blinding helps in removing the subconscious bias. By minimizing the sources of bias, the blinding studies provide a more accurate picture whether the new treatment is truly effective. Now we are talking about bias throughout. Let's spend a couple of minutes to know what a bias is. Bias is the systematic error that leads to distortion of true treatment effects. Bias in clinical trials can significantly skew the results, leading to misconclusions about the effectiveness of the treatment. There are different types of biases. Let's see what a selection bias is. Selection bias occurs when the participant enrolled in the trial is not a representative of the target area of treatment, meaning the subject selection should meet my inclusion and exclusion criteria for the study. For example, a trial testing new diabetic medication recruits only patients with controlled blood sugar level, excluding those with high complications. Next is performance bias. Performance bias arises in differences in how the participants of each group are treated beyond the intended intervention. What it means is when one group of participants receives more care and attention from the investigator if he knows which is the control group and which is the experimental group. This difference in care leads to performance bias, where one group of patients might perform or behave better. Next is detection bias. This happens when the outcomes are measured differently among the groups, affecting how the results are interpreted. This is all about misclassification of the data. Next is attrition bias. This happens when the participants drop out of the trial unevenly between the groups. Then is reporting bias. It happens when statistically significant or positive results are more likely to be published than non-significant findings. So how do we minimize this bias? Researchers constantly employ various strategies to reduce this bias throughout the trials. Some of them are randomization, blinding, intent to treat analysis, and pre-registration trials. By understanding the types of bias and methods to address them, one can gain an accurate understanding on the true effectiveness of the treatment. If you like this video, please share and subscribe to our channel CliniLaunch Knowledge Pod for more such videos. Thank you, and I will see you next time.